Anna Farago. I'm an instructor in medicine at Harvard Medical School. So today I talked about our research in small cell lung cancer. I had three different parts of my talk. The first part was focused on models that we've developed to better study this disease, and specifically what we've done here at MGH is we've developed patient-derived xenograft models of small cell lung cancer, both from biopsy specimens from patients as well as from circulating tumor cells from patients, and we've been able to over the past several years generate over 40 models and have corresponding clinical data. And I think excitingly we've had several patients from whom we've been able to develop models at serial time points, so we can start to study how the disease evolves over time in these models. And we validated that these models faithfully recapitulate the histology and the genomic features of small cell lung cancer tumors in patients. The second part was focused on a clinical trial that we have led here at Mass General, which is a trial combining the PARP inhibitor olaparib with the alkylating agent temozolomide in patients with small cell lung cancer who have been previously treated. And I showed data that we had presented previously this year at AACR, where we showed a phase 1 response rate of 46% among 13 patients in a dose escalation portion of the study, and we're now moving on to a phase 2 portion of that study. What's nice is that we've been able to generate PDX models from patients on that study and show that the patients who were initially sensitive to the combination of olaparib and temozolomide had corresponding PDX models that were also very sensitive, and then when the patients developed relapse disease, PDX models developed at that time point were much more resistant to olaparib and temozolomide. So we can model the behavior of the disease in the patients now in mouse models. And we can use that to try to identify biomarkers of sensitivity or resistance, which we hope will eventually translate into prospective biomarkers in clinical trials. And then in the last portion of my talk, I mentioned some interesting and exciting new data from AbbVie Stemcentrx with a drug that they've developed called rovalpatuzumab tesirine, which is an antibody-drug conjugate that's directed against DLL3. And I reviewed some phase 1 data that they had presented at ASCO last year, showing a response rate of about 38% among small cell lung cancer patients whose tumors express high levels of DLL3, and described the ongoing clinical development of that drug. So we look forward to seeing some of those results hopefully in the future. So I think that was the goal of the presentation today was to really cover several topics in small cell lung cancer, but really send the message that this is a disease that has typically been seen as fairly uniform, and clinical trials have generally been designed without any specific molecular selection criteria or even stratification factors. And I think what we're starting to see emerge is evidence that, in fact, small cell lung cancer is heterogeneous, and there are likely different molecular subtypes of this disease. And as we start to understand them better, we may be able to design better trials that are specifically focused on particular molecular subsets of patients, hopefully with improved outcomes based on that selection criteria.